really done some amazing work, and they made up their whole way of doing it, and it's working. It's getting incredibly valuable results. So I'll let Jessica describe that. Here's Jessica.
Thank you, Howard. Well, thank you, everyone, for being here today. It's an incredible turnout. Thank you to Susan and Howard for their efforts to put this together. Thank you for whoever made those oatmeal cinnamon cookies. One of the best cookies I've had in a while. So I'll be talking today on endangered feces, how southern resident killer whale scat samples give scientists critical health and population data to this endangered population. I think it's fair to believe that everybody here is familiar with the whales, the southern resident killer whales, a lot of the natural history. So I'm going to dive right into what we're doing, essentially, because there's so much great data that we're getting from this study, and I think to take the time to really talk through that will be really interesting to all of us. As many of you are likely familiar, the southern residents were put on the endangered species list in 2005. Following that, there was a southern resident killer whale recovery plan. And a recovery plan is put together for all endangered species. And this is done as a way to look at how to rebuild the population and promote mitigation to get the population back to where is more ideal. The southern residents in particular, there were 4 proposed threats that were outlined in this recovery plan. The increased boat traffic, decreased prey, excessive exposure to contaminants, and to the oil spills. Each of these different threats, the excessive boat traffic and the prey, the contaminants, and this threat of oil spills, are something that our study is really unique, that we can look at all 4 of these proposed threats in one study using one single type of sample. And sure enough, that type of sample is whale poop. This here is the endangered feces, scat, feces, poop, crap. There's a few other ones I could go through. But I'll use a lot of those words, except for maybe the crap one, interchangeably throughout my talk. Feces and scat it's a real treasure. It's an underappreciated resource. You'd be amazed at how much information you can get from a scat sample. From a single scat sample that's collected on many of these different endangered species that are studied in the Conservation Center for Conservation Biology, from each scat sample you can get DNA, you can look at actual individual, you can have a unique individual assigned to each scat sample, and that's something that's done with the killer whales. You can also confirm species, and you can look at sex. We can look at hormones, including stress hormones, so physiological stress, psychological stress, reproductive status, nutritional stress. You can look at pathogens, you can look at immunoglobulins, these are antibodies, which is a marker of immune status. You can also look at diet, what have they been eating, where have they been, what habitats are important for their diet, as well as toxins, looking at what toxins are they excreting as a sign of what have they been exposed to. So I'm thinking about this amazing resource of scat and all this great information you can get from it. The question must come to mind, well, how the heck do you find killer whale scat samples? Well, of course, we use a conservation canine. This is Tucker. Tucker is as hardworking as he is handsome. He's absolutely amazing. Tucker is a scat detection dog, and all of these scat detection dogs, these conservation canines, have a very strong ball drive, we call it. They love their ball. They're trained similar to narcotics dogs, drug dogs, where Tucker has shown that if he finds scat from an endangered species scat, he can associate finding that scat with getting to play with his ball. Absolutely amazing and adorable all at the same time. The last 2 years, Tucker has been handled by Elizabeth Seeley, who's been working really hard at understanding the subtleties of how Tucker recognizes and reacts to different changes in the smell and helps us guide our drivers. Carrie Koski and Deborah Giles, the last few years, have been our drivers, and how to get the whole team up to where the sample is so we can scoop it up. Prior to that, it was Catherine Ayers and Heath Smith, and of course Sam Wasser, the PI in the study, who've been working to really help Tucker be all he can be and find these samples. Tucker is amazing at what he does. Last year, we spent 495 hours in the water and found 167 scat samples. And these are highlighted right here. All the samples found in US waters are the ones, the red stars, and all the samples found in Canadian waters are the ones with the green stars. Not all of them are represented here, but most of them are. So it kind of gives you an idea, too, of where the whales have been, where we're with the whales and collecting these samples. In 2011, we spent about 411 hours in the water and got over 100 samples. So the study has had a lot of successes in these last many years, gathering quite a bit of samples on these whales. Something I'd really like to point out, because it's another great strength to this study, is recognizing the Be Whale-Wise guidelines, which is to be further than 200 yards from the whales at all times and over 400 yards out of their path. And this graph right here represents the samples that we collected in 2011. We had just about little more than 2% of our samples were collected within that 200 yards, and that was because the whales flipped on us, so we weren't able to get out of the way fast enough. We are under permit, so it's okay, but we really do our best to stay out of the way of the whales. And you'll notice here that over 80% of our samples were collected 400 yards or greater away, which is a quarter mile, which is kind of our sweet spot. We try to hang out at that quarter mile mark and just follow downwind from the whales and wait for Tucker to kind of perk up and let us know that there's a sample nearby. So we spend 495 hours in the water and we get all of these great samples. Should I warn anybody about this slide? So then what? We've got all these great samples. We want to get some great data out of these samples to help explain what's going on with these whales. So we take these samples and we'll put them in the freezer and then back during the summer, and then in the wintertime we'll take them back to Seattle, take them to the lab, and we'll start processing these samples. We'll weigh them all out, we'll take a certain weight, put them in each of these test tubes, we'll add different solvents to these test tubes that pull out the toxicants and the hormones, which are the two main measures that we're using to understand what's going on in this population and what information that we can get from these killer whales. So to go back to where we started, now that we understand that we're out there on the water, we've got this great dog, we're collecting all these great samples, how are we using these samples and this data that we're collecting to address these questions? The first threats I'm going to talk about are the top two, the increased boat traffic and the decreased prey. And this was the dissertation of Catherine Ayers, who started this project. She recently successfully defended her thesis, or dissertation, so I guess it would be Dr. Catherine Ayers. And her data has been submitted, and it's in review to be published in PLOS ONE, which is a scientific journal. So for those interested, I definitely recommend keeping your eyes out for that. It's a really great paper. And I'm going to discuss some of her findings and her data today. Catherine's approach to using these samples to address these proposed threats was by looking at hormones that she could measure in the samples. The first hormones I'm going to talk about that she looked at were these glucocorticoids and these thyroid hormones. The glucocorticoids are stress hormones. They look at psychological stress and physiologic stress. They work really quickly by mobilizing glucose, mobilizing sugars, to kind of give you that fight or flight response to something. Whereas the thyroid hormone, that's more like a basal metabolic rate. That's, in general, how are you doing? How's your nutritional status? How's your nutritional stress? If you're eating a lot, your metabolism's higher, your thyroid hormone is higher. If you're eating less, all of your metabolism goes down a little bit more. So your metabolism is slower, usually a reflection that you're eating a bit less, and everything just slows down to help make most use of what you are getting. So in consideration that she was looking at these top two threats, increased boat traffic and decreased prey availability, using these two together really helps tell that story, because the stress hormone can tell you about physiologic stress and nutritional stress, whereas the thyroid hormone really gives you more of an idea of what the nutritional stress alone was doing. And what she found is really fascinating. Looking at this physiologic stress, we have, I'm going to walk you through this for a second, because it took me a second the first time I saw it. So just focus on the green circle for a second. This x-axis here looks at the Fraser River Chinook salmon. These are the number of salmon, the counts of salmon. And it gets higher as the line goes up. This x-axis is the stress hormone, the glucocorticoids. So as it goes up, the stress goes up. So looking at these 3 years together, scat samples collected from 2007, 2008, and 2009, you can see as the salmon count was higher, the stress level was lower. And likewise, when the stress level was higher, the salmon level was lower. So it's pretty interesting, and it kind of makes sense. When you're hungry, you're stressed out. When you're not hungry, you can kind of handle everything a little bit better. Likewise, she looked at the boat traffic around the whales, on scene with the whales. And this is boat data that was collected by the Soundwatch, the boater education program. And they're out on the water every day collecting boat counts around the whales every half an hour. And using the same type of metric, you can see that as boat traffic increased, well, let's look at 2008 and 2009 first. As boat traffic increased, the stress level didn't do quite as much. You can see a little decline, but it's not statistically really telling you much. What's really noteworthy about this is 2007. You can see as the number of boats increased, the stress level really increased. So I want you to keep that in mind as we go over these next couple of slides, because this is pretty important. The next thing she looked at was this thyroid hormone, the one that looks at basal metabolic rate, understanding how much you're eating, how much you're not eating. And looking at that by day of the year, she was able to show that in early season, so spring, the spring data that she collected, the thyroid level was higher, whereas later in the year, as spring or summer and then fall came on, the thyroid level was a bit lower. So this shows us that in the early spring, the whales were in a better nutritional state, had better nutritional status than they did later in the year, because their metabolism was higher. It was metabolizing what is likely more food and nutritional availability. And this was consistent for all 3 years, which is interesting. Now to look at all of this data together, we're going to start this one by one. The line on top is, again, the Fraser River Chinook salmon. This is catch per unit effort. That's what the CPUE stands for. Now looking at 2007, 2008, and 2009 by days of the year, if we look at these salmon runs, the one that really stands out to me right away is 2008. It's got one really tall peak. So the first impression might be, wow, 2008 was a great year. But if you look at it closer, you can see that 2008 was tall, had a high peak, but it was a skinny peak, which means there was a little bit of time where there are a ton of fish. But that doesn't necessarily mean that over the course of a season that there's always going to be, dependably going to be more salmon available. Whereas for 2009, the peak's lower, but it's pretty flat. You know, there's a lot of time, there's maybe a certain amount of fish available for a longer period of time instead of a ton of fish available for a short period of time. Now, using those thoughts for 2007, you can see it's a short, skinny peak. So this kind of demonstrates that 2007 may not have been the best salmon year. There's times when there just wasn't as much available. Now, if we think back to that slide, looking at how boat traffic showed a stress response only in 2007, we may start really start to think about, as there was less fish available, that may be part of the response to the boats. Next, looking at just the boat traffic, you can see it's pretty similar, 2007, 2008, and 2009. It starts low, then around that July, August time, there's a ton of boats in the water, and then it kind of peters off again. Consistent all 3 years. Looking at the stress level for the 3 years, this is kind of a different way to look at the data I showed earlier. You can see the whales come into the Salish Sea area with a moderate stress. Their stress really dips. They have their lowest stress level kind of around this July-August period again. What's interesting, this vertical red line is when the stress level is the lowest. And if you follow that red line up, you can see that's just before the peak of the fish. And if you think about it, this fish data, which I didn't mention before, but the fish data is collected in the Albion test fishery data, which is just past Vancouver. And through calculations that Catherine did, as well as accounting for the speed the Chinook salmon can swim, it's about a 10-day lag. And sure enough, this vertical line is about 10 days short of the peak. So it kind of demonstrates that, again, when the stress is lowest is about when the fish prey availability peak is the highest. Another thing that you can really notice on this is 2007 again, it was a little different. The stress level overall was higher. It was actually higher than almost the entire season for the other 2008 and 2009. So something else was going on in 2007. The last thing is this thyroid hormone, which again is a different way to look at the data that I showed before. But you can see that when the whales came in early spring, the thyroid level was the highest. The whales were in their best nutritional state. Then their metabolism slowed down a bit, and then there was this other little blip here at the end. And as I mentioned before, the thyroid hormone was a little slower to react. So this is likely a demonstration, a reaction to this peak in the fish. So the fish get a peak, their metabolism speeds up again in response to that peak, and then it dives down again. So it's really interesting to look at all of this data together. So in thinking about it, what does this data tell us? And what Catherine came up with as some of her main findings was that in general, the fish availability, the Chinook salmon rod, had more of an influence overall on the whales than the boat traffic, except for those times when the fish were lower. When the fish were lower, when the whales did not have as much prey availability, they were more likely to have a stressful response to the boats, which makes sense. When you're hungry, you get crabby easier. Right? When you're well fed, you can handle some of those external stressors. Another thing she was able to demonstrate was that the stress level does indeed show short-term response. The glucocorticoids show short-term response, and these thyroid levels, they are more reflective of this longer-term response. Her data did a great job of showing that. Her next conclusion was describing that the southern resident killer whales, they arrive in their best condition of the year. And that was demonstrated in looking at the different thyroid hormone levels. And that led her and Sam and others to really start to think about, Sam is the PI, Sam Wasser, led them to really start to think about the importance of early season prey availability. So that may be those early Fraser River Chinook salmon, which is a predominant diet for the whales during the summer months, or possibly start to really think about the Chinook or the Columbia River Chinook salmon and other fish that these whales are eating earlier in the season, and how they may be really, really influential. They may be really important to their health overall. And that's what this bullet on the bottom really demonstrates. One last thing I'd like to point out before moving on. I'm going to go back to the slide for a second. Talking again about 2007 being a year where there's a little more stress in general. Maybe not as much fish, a little more stress was demonstrated. Something really interesting that at this point is still anecdotal, but the winter following 2007 was shown to have the highest mortality, winter mortality. The most whales went missing that winter. And that includes, and I believe it was also 2 reproductive age females were a part of that. So it's pretty interesting. The same mortality losses were not seen in the other years, and that's still being investigated, but it's just something interesting to point out. One other thing that Catherine looked at for the hormones that she looked at were reproductive hormones. This is essentially a pregnancy test for whales using poop. I don't know if this would market very well in the shelves or grocery stores, but it's still a bit in the works, but there's some really interesting data to show you. And ultimately, the hope is to use this to look at winter reproductive success. At this point, when a whale comes back in and they do not have a calf, we don't know if they just weren't pregnant, if they were pregnant and were not able to carry the child to term, or if they had the child and the child didn't make it. At this point, the calf did not make it. At this point, it's not known. So what Catherine looked at were progesterones, where she was able to take all fecal samples from female whales and break it into 5 categories. Juvenile whales, so non-reproductive age, reproductive age with no calf, reproductive age in pregnant, reproductive age in lactating, or post-reproductive. And it's pretty clear that there's a pattern going on here. The reproductive age females have a lot more progesterone than the others. The other thing she looked at, which may be surprising, is testosterone. It's not uncommon for certain mammals, female mammals, to have higher testosterone levels. And looking at these same categories, you can see there's also a similar pattern. You may note the y-axis on each of these. This is 10,000 and this is 30,000. So the progesterones are far greater, but there's still a pattern within the testosterone that's worth looking at. So Catherine took this, thought about it, and assigned thresholds to say that when a female whale, or when a whale has more than 1,700 nanograms per gram of progesterone, that whale is likely a female. And likewise, when they have more than 50 nanograms per gram of testosterone, that whale is also likely a female as well. And using this combination of hormone measures to help assign the sex to the animal. And this was used for the extent that the genotyping is able to be used at this point, which is for over 60% of the whales have a specific genotype assigned to them. So our scat samples can now be assigned to a unique whale, which is so cool and so amazing. But for those that don't have a unique genotype at this point, this is something Catherine developed to be able to at least assign a sex, to know is it male or is it female. And what she did is took this progesterone and testosterone and put it on a graph together. And what she found was all whales, all samples, everything over 50 nanograms per gram of testosterone, but below this 1700 threshold, they were all the male samples. She then looked at everything that was above the 1700 progesterone threshold and the 50 testosterone threshold, and they were all the reproductive age females. And everybody in this didn't fall into those categories, just didn't get assigned anything. So this is still in development, but it's really exciting. She has been validating an estrogen hormone assay to add on to this. So we'll have progesterone, testosterone, and estrogen. And together, the hope is to create what will be the pregnancy test. At this point, we can confidently assign sex, but next will be also to confidently assign pregnancy status. So now I'm going to shift gears a little bit. Everything I've talked about so far has been the work that Catherine did, the work she did with the hormones, the work she did on her dissertation. Next, I'm going to address these other two proposed threats. This is the excessive exposure to contaminants and risk of exposure to oil spills. And these are the two that are part of the project I'm working on and will actually be my dissertation. So to give you a bit of a background, I don't know if you've heard these terms of the toxic whales, to understand what that really means. So I'm going to give you a bit of background on what these toxins are. There's three main classes of compounds that are studied for toxicity within these whales. The first are these organochlorinated pesticides. So they're pesticides with chlorines, basically. These include DDTs, dieldrins, lindanes, a lot of things. Most, dare I say all, have already been banned, at least in the US. They're all pretty nasty. They've been shown to have some pretty bad health side effects. They're biopersistent, they're bioaccumulative, they don't go away. And these are compounds that are still being measured in killer whales as well as other parts of the ecosystem. A second one are these PCBs, or polychlorinated biphenyls. These have also been banned in the US. They were banned in the late 70s. They are compounds that are used in industry for transistors, for capacitors, for industrial lubricants that could handle high heat. Also really nasty. Also bioaccumulative, also biopersistent, also resistant to degradation. The PCBs, there's actually 209 of these, which is kind of amazing. There's one parent compound and then 209 versions, variations of it, all of which are nasty, but there's different reasons that each can be bad. And then the third one are PBDEs, these polybrominated diphenyl ethers. These are the flame retardants. These are reasons couches don't burn anymore, or homes don't burn down. They're in baby cribs and in computers and in the chairs you may be sitting in. They have padding. They're in everything. And these actually have not all been banned. There's a real push to get them banned at this point because they've been shown to have a lot of similar nasty side effects. The PBDEs also have 209 different versions, all that have the same type of parent compound. So all three of these, they all may be really different, like agricultural, industrial, and consumer products. They all have different sources, but they're all structurally, chemically pretty similar, which leads them all to have this bioaccumulative, biopersistent characteristic, as well as being lipophilic. They love lipids, love fat, so they're all stored in the fat tissue within us, within the whales, within all other organisms, which is part of how they don't go away. They're just constantly cycling through the ecosystem. So how these three got to be the ones that got the most attention in the whales are due to past studies that have looked at blubber biopsy samples in these killer whales. This was a study that looked at PCBs. These little pictures will follow along, so you don't have to pay attention to the acronyms so much. It looked at northern, southern, residents, and transients. And you'll notice the levels are a bit different. Any thoughts as to why? I heard diet. As many of you do know, the northern and southern residents are fish-eating whales, whereas the transients are marine mammal-eating whales. And so they're one more step up in the food chain, leading to them having higher exposures and higher vulnerability to bioaccumulate these toxins. The reasons the northern and southern residents are a bit different leads to, it has been shown that the prey source of the northern residents is generally less contaminated. And if you think of it, they just hang out in less urbanized areas. The whales here are in this Puget Sound area that has just a lot of urbanization and industrialization that surrounds it. There's something else you may notice about this. Yeah, any thoughts as to why the males and females are so different?
No lactation.
Yeah, I heard a lot of very correct answers. Maternal transfer. It's transferred through the placenta and as well as through lactation. So it's actually shown the firstborn of the female whales, their levels generally land somewhere between the male and the female levels. And then all other calves are also vulnerable, but not as much as that firstborn. So in talking about all the similar chemical characteristics that these toxins have, they also all have similar health endpoints. They have all been demonstrated to be reproductive toxins, immunotoxins, and neurotoxins, which are awful to think of this endangered population being exposed to high levels of compounds that have these effects. The health endpoint that I'm really going to look at in my research is the reproductive toxin endpoint. If you've heard of the term endocrine disruptor, all of these compounds are considered endocrine disruptors. Endocrine is hormone, so disrupting physiologic levels, physiologic regulation of hormones. Hormones are really important. They drive everything that goes on in our body. And these compounds disrupt that, and that can lead to a lot of nasty side effects, including reproductive toxicity and the inability to appropriately reproduce at a level that you would if you did not have the toxin load. In past studies as well, the whales have been shown to have levels of these compounds, this is PCBs again, that exceed a health effects threshold. So it has already been demonstrated that these whales have levels that are beyond what is considered healthy. And so it certainly leaves reason that we should look at this further to better understand what's going on and how we can help with remediation. So what we're doing in our study is we're taking these same samples, the exact same samples that Catherine has done all of this great hormone work on. We're using those same samples to look at toxicant levels in the whales. So far, we've only done 2. However, that's really exciting. We've done 2 and it's been successful. So what I'm going to be showing you today is our preliminary findings from our test samples, and I'm going to show you what we're finding and show you that it's working. So the exciting part will be the talk next year, or years to come, where we'll be taking hundreds of samples and give you a bigger picture idea of what's going on. But for now, I can show you that in these 2 samples that we've used as test samples to test the methodology, one was an L-pod sample and one was a J-pod sample. And the first set of data that I have here is DDT levels. DDT, banned in the late 60s. But what you can see is this, both have levels of it, but this L-pod whale has more than twice the level of the J-pod whale. And if you've ever heard of this California signature, has anybody heard this term? It's a term that's been used to describe the ecosystem and different organisms off of California that have been exposed to high agricultural impacts as well as past spills, specifically with DDT, giving these organisms off of California this special signature. And it's my understanding L-pod has been seen off of the coast of California much more so than J and K-pod. And biopsy samples have shown this a bit as well. So what this shows is something's working because our L-pod sample had a lot more DDT than the J-pod sample. We also looked at PCB levels. This right here is 40 of the 209. I mentioned there's 209 different PCBs. This is 40 of them added together. And the levels are pretty similar between the 2, which is expected. And both are pretty high, which is not great for the whales. And then last are the PBDEs, which again are at measurable levels. And with all toxicology, the dose is the poison. So just because these levels of PBDEs are less than the PCBs doesn't mean it's not as dangerous or scary. It just means that there happens to be less. So to take a bit of a closer look at this, these are all of those other chlorinated pesticides. I'm not going to spend a ton of time on this slide. I just want to show you that the data's here. These are the non-DDT pesticides, the dieldrins, the lindbanes, all that other stuff. And you can see there's some pretty high levels of these compounds found in both whales. What I would like to take a few more seconds to talk about is to look at these PCBs in a little more detail. These are all 40 lined up, and you can see different congeners are called, different versions of this PCB are higher than others. And to compare this to the levels that have been found in blubber biopsy samples, we can see that comparing, this is PCB 101, PCB 138, and PCB 153. The three highest in the scat sample were also the three highest in the blubber biopsy sample. So this shows us it's working. Something else that's exciting about this from a scientific standpoint is these are the three of the six what they call indicator PCBs. They're signs of environmental exposure, of biologic relevance, and give you a sign of all other PCB exposures. So these can be used to help really tease out some of the biologic effects, the adverse health outcomes associated with exposure to any of the PCBs. To do the same thing with the PBDs, this is 11 of the 209 that we found in the scat samples. And to compare that to demonstrate how that looks compared to blubber biopsy samples, you can see this is PBDE 47, 99, and 100. So again, the three highest that we saw in the scat sample were the three highest that were also found in the blubber biopsy sample. So what? So we've shown that these whales are contaminated, we've shown that they're high levels. How do we actually show that the contamination that we see in these whales can be related to an outcome measure of some sort, show that there is an adverse effect? I'm going to show you mock data, and I apologize, but I think it helps visualize what the plan is. But in the discussion of these being reproductive toxins, endocrine disruptors, what I'm expecting to see is something like this. I'm expecting to see the reproductive success of these whales is going to decrease as the toxin level is going to decrease. So the less reproductive success when there's higher toxin levels. And when there's lower toxin levels, you're going to have better reproductive success. And this is the type of thing that we're going to look at once we're able to do, which we're in the process of doing, is looking at the toxin levels in all the scat samples that we've collected. And we'll relate that to these different measures. I'll also do something similar looking at the hormone levels, pulling in Catherine's work and the hormone work that she did with the thyroid hormones and the stress hormones and these reproductive hormones, and relate those measures to the toxin measures. And I'm expecting to see that the more toxin, the less hormone, and some non-linear version of that from this messed up disruption. But we hope to be able to show that through the data and use it to help tease out what's going on and better understand the big story. Isn't it so sad? I was looking through Google Images trying to find some comic relief at this point in the talk. All the pictures are so sad, so I picked this one. The last thing I'd like to talk about is oil spills, which is really relevant, especially with the last talk that was just here. Puget Sound, and it was Matt and Lindsay, if you're here, correct me. Correct me if I'm wrong, I didn't hear a number on this, but in what I've read, there are 15 billion gallons of crude oil and petroleum byproducts that pass through Puget Sound annually. That's so much. Not only is that a really high number, but Puget Sound area is a confined geographic area. If there's ever a spill, an oil spill of any sort, it is going to be really catastrophic, and it's going to be devastating to the whales who depend on the health of these waters. So something that we're also going to be doing with these samples, and I've got some test data, some of the validation data to show you, is to start to measure current levels of oil spill byproducts in the whales to serve as a baseline measure. This is something that has not been available for some of the bigger oil spills that we know of, the Exxon Valdez spill, the Deepwater Horizon spill. They did not have baseline measures on the areas of that ecosystem. So for us to be able to have available some baseline measures on the killer whales, then we'll know what we're working towards as far as remediation, mitigation, cleanup, how to get back to where we started. So what we looked at are what are called PAHs, polycyclic aromatic hydrocarbons. They're byproducts of crude oil exposures. There's 16 that are considered priority pollutants by the EPA. Many of those are also considered probable carcinogens by the National Institutes of Health. So what we did is measured the 16 priority pollutants to see what the current levels are in the whales. And look at all that white space. This is good. It means that the exposure levels at this point are smaller. Many of these were not even detectable at levels that were detectable. So we'll be able to take these measures, measure in all the samples we have, and it'll offer a baseline measure in case something does happen. Lastly, I would just like to talk a bit on the implications of this research. Of course, as I keep bringing up, this will offer data. It'll offer resource management for government, regulatory, community organizations. The recovery plan acknowledged there's data gaps out there. And what this study can do is look at all 4 proposed threats and help answer the questions for all 4 of those using the exact same samples, using one sample to address all 4 questions for across the seasons, across the years. And this is not only great for killer whale conservation, but also for ecosystem restoration and for salmon recovery efforts to really keep Puget Sound as healthy as it can possibly be. Next, another great part of the studies is longitudinal, where we have high sample numbers, which is something many other studies have struggled with because it's a tough population to study. But we have, I'm plugging Tucker again, we have him. We have these conservation canines where we can hang out a quarter mile away from the whales at all times and collect these samples that are giving us a wealth of information. And by collecting early, peak, and late season samples over many, many years, we have 2 more years of funding at this point, and the study started in 2006. It's a lot of data. You can look at different environmental factors over those years, different combinations of effects that can help us understand the data we have to help us understand what we can do with it. Next is the baseline for mitigation success, not only with the oil spills, but also for cleanup, for these toxin measures, these PCBs, these PBDEs, for new vessel regulations, for using the 200 yards instead of 100 yards. We have stress levels that can show what's that is doing from the killer whale standpoint. And last, this is my favorite, is the outreach value. Education, encouraging good stewardship practices, this is where it all begins. And this study is great because we have Tucker, we use poop, and it's just, it gets people to ask questions. But this is where it involves all of us, because it's the community in general, as a greater whole, understanding what's going on, how we're part of it. We're the ones who are doing the polluting, we're the ones who are encouraging some of these practices. So the better we understand the issue, the better we can help be a part of the solution. And then future work, briefly, is to really look at these toxins a bit more, see how it relates to fish beak. As they're hungry, will they start to metabolize some of these fat stores, which release more toxins into their system? The endocrine disruption, like I talked about before, we'll look at immunosuppression. One of those other toxic effects, the health outcomes, was immunosuppression. So we can maybe we're looking to evaluate antibodies to understand immune status in relation to toxin levels. This is linking the pressures to reproductive failure. This is not just with the toxins, but with all the hormone data as well. And then ideally, we could come up with the funding to get some scat samples on transient whales and the northern resident whales and really use these other ecotypes to compare the values we've got and see how it all interrelates. Lastly, I would just like to offer some acknowledgments. There's so many people involved to make this study work, and this just does not cover it. Of course, we have our funding. The Washington Sea Grant has been funding us for 4 years total now. We have some really important collaborators at NOAA with Soundwatch and the Whale Museum, the Center for Whale Research in Cascadia, and then of many, many individuals and groups. Giles and Carrie have been driving for us these last few years. Liz has been such a wonderful dog handler with Tucker these last few years. Liz, who has donated the use of her boat in kind. Lynn Miner, who has let us use open the doors of her house to our team and our interns, just really helped with the community of what we do, as well as financially. numerous field assistants, the Conservation Canine Program, Center for Conservation Biology, the Whale Museum, Course Orca Network, the Wasser Lab, National Marine Fisheries, who gives us the permit, Just Sail, who's given us a couple of great interns. This is all of you guys. This is my boyfriend. And many more. We have a great Facebook page. We have a really great website. Please write these down and go check them out. And this is my email address. If you have any follow-up questions or anything, just don't hesitate to contact me. Thank you.